Hi, everybody, and welcome to the online version of Biochemistry 521, General Biochemistry. So, this is a one semester course designed to introduce you to the principles of biochemistry. My name is David Meekins, and I'll be your instructor for the course. So, I got my PhD in biochemistry, and I worked for 12 years as a biomedical biochemical researcher before switching gears and becoming an instructor. So please also take a look at the syllabus video that I posted. So that's going to go over everything on the syllabus, including things like expectations, the grading breakdown, and also the schedule for the course. So please take a look at that before you start launching into the lectures. Now, today's lecture will be lecture number 1, an introduction and aqueous solutions. So I'll first start with going over what is biochemistry. What does biochemistry mean as a discipline within the sciences? I'll then move on to discuss aqueous solutions. So basically all of biochemistry happens in aqueous solutions. So we have to have some understanding of what water is and how it interacts with other molecules. And lastly, we'll have a discussion on some of the weak non-covalent interactions that are important to biochemistry. So with that, let's launch into the first lecture topic, which is what is biochemistry? So the first topic we're going to talk about today is what is biochemistry? So put simply, biochemistry is the study of chemical processes involved in life. So bio means life, and chemistry, of course, is just the chemistry involved in sustaining biological systems. So when you think of biology, you might often think of higher organisms and the ecosystems, which is the interaction between different organisms and their environment. But if you dig a little deeper, you could also see that those organisms and ecosystems are built up by individual cells and collections of cells called organs and the interactions between those within an organism. Now, biochemistry actually goes even a little deeper. So we're going to be studying atoms, molecules, and the interactions and the atoms and the chemistry that basically sustains cells, organs, and ultimately living things. So different species, humans, and all that sort of stuff. So on the left here, we have some examples of different molecules that are necessary to sustain life. So right here, for instance, we have a molecule of ATP. And so that's one of the main energy molecules that allows people to race in bike races like you see over here on the right. We also have a molecule tetrodotoxin. And so that is actually a potent toxin that's produced by pufferfish to keep predators at bay. And we also have a molecule down here in the right, which is hemoglobin, which allows your blood cells to store oxygen and to carry it to different parts of your body. And biochemistry is also very important in has a lot of medical applications. So for instance, if there's a mutation in your hemoglobin protein, it can cause sickle cell disease, which is shown right. An example is right here where your cells aren't able to effectively carry oxygen. And this can lead to disease in humans. So overall, biochemistry is the study of chemical processes involved in life. And it's very important for things like agriculture and medicine and nutrition. And so it'll be very important to any further studies that you have down the line. So there are 4 major classes of biochemical molecules that you're going to need to know and that you're going to come across over and over again in the study of biochemistry. So pretty much all of biochemistry is built on these 4 classes of biochemical molecules. So the first one is proteins. So proteins are polymers of amino acids folded into precise 3-dimensional structures. So over here, you can see we have individual amino acids, which are just chemical groups. These amino acids are then linked together in a specific sequence of specific amino acids. And that specific sequence of amino acids dictates how that amino acid chain is going to fold into a 3-dimensional conformation to form a protein. And the function of that protein within your body is going to be dictated by that 3-dimensional folding. So proteins do a lot of different jobs in your body. For instance, they catalyze chemical reactions, so they can change one chemical into another or combine two chemicals, such as with joining amino acids into a specific sequence. And proteins that catalyze chemical reactions are called enzymes. The proteins that also provide structural support. They also perform signaling molecules that allows your cells to communicate with one another. And they're also involved in mobility if you think about your muscles or the flagella in a bacteria. So proteins are very important to biochemistry, and we'll talk a lot about proteins throughout the course. The next class of biochemical molecules are called nucleic acids. So you're probably most familiar with these in the form of DNA or RNA. So our DNA and RNA are basically polymers of nucleotides, which are nucleic acids. So the function that you're most familiar with is probably in the form of DNA. So here we have a depiction of a DNA double helix. And so this is, again, a polymer of nucleic acids. And so DNA stores and conveys genetic information. But nucleic acids also do other stuff in your body, including they're involved in energetics and metabolism. So you have a molecule here of ATP, which I mentioned is the major source of energy in your body. Now, the next class of biochemical molecules that we'll encounter are lipids. So lipids, which you probably know as like fat molecules, but there's so much more than that. So lipids are hydrophobic hydrocarbons linked to a hydrophilic head group. So as you can see, here's a depiction of a lipid molecule in a space filling model. So you can see you have this hydrophobic tail over here with a hydrophilic head over here. And this is often shortened or made simpler with this diagram here right next to it. Now, lipids are very important to life because they are what form cellular membranes, which keep the inside of the cell at a different environment than the outside of the cell. And you can see membranes are formed by this sort of lipid bilayer where the hydrophobic tails interact with each other and the hydrophilic heads interact with both the intracellular environment and the extracellular environment. But like I said, lipids are even more than that. And for instance, this steroid molecule here is also considered a lipid. So lipids do a lot of stuff in the body. Like I said, they're involved in cellular membranes. They're also involved in energy storage. So fat molecules in your cells are lipids, and those are broken down for the use of energy. They're also involved in cell signaling, like for instance, these steroid molecules. And they also are involved in protein modification that allows proteins to do lots of different things. And the fourth major class of biochemical molecules are carbohydrates. So you're probably most familiar with these in the forms of bread and pasta. But carbohydrates are literally carbon hydrates. So they have the chemical formula CH2O at a different with an N group. So multiple copies of this same chemical formula, CH2O. And these can form large polymers. So you can see here's a glucose molecule, and you can sort of see that CH2O chemistry here. So there's a CH2O, CH2O, and so on and so forth. Now, like I said, these can form large polymers, such as this shown right here, which is glucose, which is involved in energy storage. And carbohydrates can also perform structural roles, such as this molecule here, which is cellulose, which provides structural support to plant cell walls. So carbohydrates can also be components of proteins, so they can modify proteins, and they're also part of nucleic acids and often parts of lipid molecules as well. So those are the four major classes of biochemical molecules: proteins, nucleic acids, lipids, and carbohydrates. So another topic I want to touch on is the central dogma of biology. So this is critically important for understanding how biological systems work. So the central dogma was first proposed by Francis Crick, who was one of the four co-discoverers of the structure of the DNA double helix. It was first proposed in 1958, And it describes this cellular information processing scheme that's fundamental to all biological systems. So I had a previously, and when I was an undergrad, I had a professor who basically said that the secret of life is to get the right protein in the right concentration to the right place at the right time. So the central dogma is sort of how this is achieved. So put most simply, it's DNA to RNA to protein. And you can see, so if you can remember that, you're going to be in good shape. But if we look at it a little more detail, we have DNA molecules, which are the main information storage system in your bodies. And DNA molecules, of course, can replicate, as you can see here. And then that DNA is transcribed. So through a process of transcription, that information in the DNA is transcribed into information in RNA. And then that information in the RNA is translated into a specific protein molecule. So throughout this course, we'll look at this in much more detail. So we'll look at actually the biochemical mechanisms that are involved in DNA replication and then transcription of RNA molecules from the DNA double helix, and then also translation, so how that RNA molecules are translated into specific protein molecules, which I mentioned are polymers of amino acids. So the central dogma, DNA is transcribed into RNA, and RNA is translated into proteins, is going to be a very important topic for this course. So another topic that we're going to cover a little bit later in the course is about cell signaling or signal transduction. And this is one of my favorite topics in all of biochemistry. It's just simply fascinating. And it explains a lot about, if you think about it, about how your body works from day to day. So cell signaling, also called signal transduction, it describes how physiological stimuli are received by cells, resulting in a cellular response. So your body has to have ways both to respond to external stimuli. So if you're walking down the street and you see a lion, your body has to be able to tell the other cells to have a specific response so you can get away from that lion. Also, if you just ate a meal and your blood glucose levels start spiking, then your body has to have ways to tell your cells to start taking in glucose and to change its metabolism in response to that. Cell signaling also is how your body just maintains its regular homeostatic state. So your blood pressure, stuff like that, is regulated by cell signaling. So it's a very interesting topic. There's sort of a diagram here showing many of the different players in cell signaling, and also a description of endocrine signaling, which is where cells from one part of the body can release molecules that travel through the bloodstream and are received and can change cells in another part of the body, resulting in a physiological response. But there's also paracrine signaling, where cells can change the behavior of or the physiological state of neighboring cells, and autocrine signaling, where a cell can actually regulate itself through the release of small molecules. So this is a topic that's pretty interesting, and we'll go over it in this course and learn a little bit more about how this process works. And lastly, we'll also spend a great deal of time in this course talking about metabolism. So metabolism is basically the chemical reactions that result in energy generation and also the breakdown and production of building blocks for different biomolecules in your body. So this process is central to sustaining life. It's very important for nutrition and everything, and also just medical sciences in general. So we'll go in a pretty big detail, great detail into the metabolic pathways in your body. So I know that looking at these just right off the bat, these sort of complicated diagrams here can seem a little daunting, but by the end of the course, you'll have a much better understanding on how our bodies take in nutrients, how they convert them into energy, and how they convert them into other biomolecules. All right, so that concludes our introductory discussion on what is biochemistry. You do not need to remember everything that I just told you, but there are some key concepts I'm going to need you to study and hold on to. So one is, what is the definition of biochemistry? So biochemistry is the study of chemical processes that are fundamental to life. Two, you're going to need to know the 4 classes of biochemical molecules. So proteins, nucleic acids, lipids, and carbohydrates. You need to know the general characteristics of each of them and how they differ from each other, and also some of their functions within the cell. And then third, you're going to need to know the central dogma of biology, not just for this course, but because it's one of the secrets of life. So you are going to need to know that DNA is transcribed into RNA, is translated into proteins. So with that, let's launch into the second part of this lecture, which is a discussion on aqueous solutions. So it's a very important concept to understand when we're going through this course is that biochemical interactions basically all occur in aqueous solutions, that is, solutions of water. So we'll have to go over some important features of water to sort of understand how this works. So one of the most important things about water is that water is a polar molecule. So water, of course, is um, formed of H2O. So as you can see, here's a water molecule here. And water is polar because the oxygen on water has a slight negative charge, which means the electrons in oxygen are going to be or in the water molecule are going to be predominantly around that oxygen molecule, giving it a slight negative charge, which is signified as delta negative. Whereas the hydrogens on the molecule are going to have a slight positive charge, which is signified by delta positive. So this polar aspect of water permits it to form hydrogen bonds between this water molecule and other water molecules. So this is sort of demonstrated here, where that slight negative charge on an oxygen atom is going to form a hydrogen bond with the slight positive charge on a hydrogen molecule of a different water molecule. And you can see that in a water aqueous solution, these hydrogen bonds are going to be forming between all the different water molecules. So this gives liquid water an ordered or cohesive sort of characteristic. And this is actually how really tall trees like redwood trees are able to bring water all the way from the soil to the very tops of the trees. Now, importantly, water can also form these hydrogen bonds with other polar molecules, such as biomolecules that are important to sustaining life. So, all different polar biomolecules are going to dissolve in water because they're able to form these same types of hydrogen bonds that water is able to form with itself. And equally important, water cannot dissolve nonpolar molecules. So, this is also an important concept for biochemistry. And one of the reasons this is so important for biochemistry is because it drives the hydrophobic effect. So, the hydrophobic effect is the phenomenon where nonpolar hydrophobic molecules will cluster together in an aqueous solution. So, we have, for instance, a molecule of benzene right here, which is a hydrophobic molecule just consisting of carbons and hydrogens. And so if benzene is put into an aqueous solution, the benzene molecules are going to interact with each other, as you can see from this diagram here. So what's causing this interaction? Well, it's actually not just the affinity for benzene molecules with other benzene molecules. Instead, this is a purely entropy-driven process. So it's a thermodynamic process driven by the laws of thermodynamics. So, if you'll remember back to probably another course that you've taken, entropy is a measure of randomness or disorganization in a system. So, increased entropy means there's increased randomness or disorder. Now, one of the laws of thermodynamics is that all systems are sort of tending towards increased entropy or increased randomness or disorder. So the hydrophobic effect is actually driven by the increased entropy of water when hydrophobic molecules associate. So this is pretty clearly outlined in this figure up here at the top. So when nonpolar molecules are not associated, these water molecules are forced to sort of organize themselves around the nonpolar molecules, as you can see here. Now, when these nonpolar molecules come together, there's less water molecules that need to organize themselves around the nonpolar molecules. And you have these molecules up here that are now free to exist in solution. So this part of the figure shows that there's an increase in entropy when these molecules come together. So the hydrophobic effect is very important, and it's important to understand that this is actually an entropy-driven process based on the laws of thermodynamics. And one of the reasons why the hydrophobic effect is so important to biochemistry is that it powers both protein folding and membrane formation. So these are both critical to sustaining life, and the hydrophobic effect, it powers both of them. So with protein folding, remember that we have these sort of chains of individual of amino acids that are linked together. So amino acids have different characteristics, and many of them are hydrophobic. So the hydrophobic residues will actually cluster towards the inside of molecules and drive protein folding, and the hydrophobic effect is what powers this. It's also important for membrane formation. So you have membranes that delineate the exterior of the cell from the interior of the cell, and that's very important for life. And this is driven by the association of these hydrophobic tails on lipid molecules in the lipid bilayer that forms the membrane. And so these hydrophobic heads will interact out here. And this is also powered by the hydrophobic effect. So the hydrophobic effect is a critical sort of process to understand if you want to understand biochemistry, especially because it powers both protein folding and membrane formation. So now we'll move into some additional non-covalent interactions that are important for biochemistry. So the first one we'll talk about are electrostatic interactions, also called ionic interactions. So these interactions occur between two molecules with distinct charges, so not the partial charges that we saw with hydrogen bonds. Electrostatic interactions occur with two molecules with distinct charges. So as you can see here, here's an example of just table salt, sodium chloride. So these sodium atoms have a complete positive charge, and these chloride ions have a negative charge. And so the electrostatic interaction is the interaction between these two molecules. Now, it's also important to know that electrostatic interactions can be weakened by water, so that hydrogen bonding interaction between water, or actually it's a different kind of reaction, but the interaction between water is able to sort of break up electrostatic interactions, which is why table salt dissolves so readily in an aqueous solution. So you can see here the partial positive charge of the oxygens on water, or partial negative charge of oxygen on water can interact with the positive charge on sodium. And you can see here the partial positive charge on the hydrogens can interact with the chloride, the negatively charged chloride ions. So this is because electrostatic interactions are greatly weakened by water. They're less common in biochemical interactions, but you do commonly find them. One example is what's called salt bridges that occur within or between proteins. So these are commonly encountered. And for instance, here we have a depiction of the human ACE2 receptor and the SARS-CoV-2 spike protein, which is the protein on the outside of the SARS-CoV-2 virion. And it will bind to the human ACE2 receptor, and that's how it gets in your cells and causes disease. So you can see there's a lot of interactions between these two molecules that allows them to interact, and it includes interaction between this negatively charged amino acid on the ACE2 protein and this positively charged amino acid on the SARS-CoV-2 spike protein. So this is an example of an electrostatic interaction that occurs in a biological system. So while they are not insanely common, you do encounter them in biology. Now, again, we'll revisit hydrogen bonding because it is a very important non-covalent interaction that happens between biomolecules. So between different biomolecules and between biomolecules and water. So hydrogen bonding is not just limited to water-water interactions. So hydrogen bonding is a non-covalent interaction between hydrogen and an electronegative atom, which is most commonly oxygen or nitrogen. So this is also very important in proteins. So you can see here's two amino acid backbone molecules in a protein. And so we have this electronegative oxygen atom forming a hydrogen bond with a hydrogen that's bound to a nitrogen atom, either within the same protein or in different proteins. And these can exchange and also form hydrogen bonds with water. But if you look at this diagram over here, you can get an idea of what types of interactions will form hydrogen bonds with other atoms. So nitrogen with hydrogen can form hydrogen bond. This hydrogen bound covalently bound to a nitrogen is going to be electropositive. So it can form hydrogen bonds with a neighboring electronegative nitrogen atom. Same with oxygen atoms. And then hydrogens covalently bonded to an electronegative oxygen atom can form hydrogen bonds with an electronegative nitrogen atom or oxygen atom. So that's very important. And these are weaker than covalent interactions, obviously, but the combination of many of these hydrogen bonding interactions can provide a lot of strength to interactions between biomolecules when they're combined over, a large molecule. And a good example of this is that many weak hydrogen bonds are essential for the stability of the DNA double helix and also of protein folding. But if you look at this DNA double helix, of course, DNA it interact it, the double helix is formed by interactions between A's and T's and also C's and G's. And these interactions are formed by hydrogen bonds. So 2 in the case of A's and T's, and 3 in the case of C's and G's. So hydrogen bonding, very important concept to understand for biochemistry. And the last non-covalent interaction that we're going to talk about are van der Waals interactions. So these ones are a little interesting. So van der Waals interactions are interactions between molecules that are not polar or charged, but that can have transient asymmetries in the charge distribution. And this can cause weak electrostatic interactions when these two molecules are at a particular distance. So you can see here we have a molecule that's not polar or charged, but at any given time, the electrons in it may be in one side of the molecule more than another side of the molecule. And this can actually cause additional transient asymmetries in neighboring molecules, causing this nonpolar, non-charged molecule to have partial charges in each side of the interaction. And then that allows van der Waals interactions between these two molecules. So it's a very weak interaction, again, like we said, but if you combine a lot of these, they can provide quite a bit of strength and stability to different biomolecules in biochemical processes. So for instance, we have the DNA double helix again. So these two nucleic acids are interact via hydrogen bonds, but in between these two nucleic acids, is sort of this phenomenon called stacking, and that is based entirely upon these van der Waals interactions. And actually, the ability for animals like geckos to walk up walls is driven entirely by van der Waals interactions. So overall, you can see that there are 4 essential non-covalent interactions that are important to biochemistry and explain how different biomolecules are able to interact with them. So there are electrostatic interactions, hydrogen bonding interactions, the hydrophobic interactions based on the hydrophobic effect, and van der Waals interactions between different molecules. So just for reference on the relative strength of each of these, so covalent bonds, which are going to be much stronger than each of these, contain 200 to 400 kilojoules per mole of energy. So that's the amount of energy that would be released if you were to break a covalent bond. So electrostatic interactions of these 4 are the strongest. So they have on average 40 to 200 kilojoule per mole of energy contained within them. Hydrogen bonds are much weaker, but the second strongest, so they have 4 to 13 kilojoules per mole. Hydrophobic interactions based on the hydrophobic effect are 3 to 10 kilojoules per mole. And then van der Waals are by far the weakest of these important non-covalent interactions, with only 2 to 4 kilojoules per mole. So even though these are weaker than covalent interactions, they're very important for biochemistry and very important for you to understand as we continue to move through the course. So that concludes the second part of our lecture, aqueous solutions. So there's a few concepts that I really want to emphasize. One is that biochemical interactions occur in aqueous solutions. So you have to understand that water is a polar molecule. Therefore, it can form hydrogen bonding interactions with other water molecules and other polar molecules in biological solutions. You also need to understand the hydrophobic effect. So this is the phenomenon where hydrophobic molecules will interact in an aqueous solution due to an entropy-driven process, and that this is very important for both protein folding and for membrane formation. And lastly, you need to remember the 3 additional weak non-covalent interactions that are integral to biochemistry: electrostatic interactions, hydrogen bonding interactions, and van der Waals interactions. So I hope you enjoyed the lecture, and please let me know if you have any questions.